Every single second, your body is churning through millions of chemical reactions. Whether you're breaking down your breakfast, contracting a muscle to walk, or firing a single neuron, life is an ongoing chemical factory. But there is a massive logistical problem at the heart of this system. Without help, the vast majority of these chemical reactions are painfully slow. If you had to wait for them to happen spontaneously without any assistance, digesting a single meal could literally take years. To bridge that gap, biology relies on a specialized class of molecules, enzymes. Enzymes are specialized proteins that act as biological catalysts. Their entire purpose in your body is to drastically speed up these chemical reactions, all without ever being consumed or damaged in the process. To understand how they do this, we have to look at what scientists call activation energy. Notice this massive, steep peak? That's the initial energy hurdle needed to start the reaction. Without help, clearing it takes tremendous effort. Now watch when an enzyme arrives. It grabs the molecules, drastically lowering that activation energy barrier. By flattening the hurdle, enzymes accelerate the pace of these processes, from impossibly slow to near instantaneous. They make the rapid-fire chemistry of human survival physically possible. When we zoom in from those abstract energy charts down to the physical reality of a single enzyme, we see a complex three-dimensional machine. On the surface of that machine is a highly specific, geometrically unique pocket known as the active site. Floating around nearby is the substrate. This is the exact target molecule that a particular enzyme is designed to interact with. Because of its unique shape, only one specific substrate can fit into one specific active site. Biologists call this the lock and key model. The geometry has to match perfectly. Once it locks in, the reaction triggers instantly. The enzyme splits or reshapes the substrate into a brand new product and releases it. And importantly, the enzyme itself remains completely unchanged, ready to grab the next substrate and repeat the cycle. This struct geometric perfection prevents chemical chaos. Even with thousands of different reactions happening simultaneously inside your cells, each enzyme acts only on its designated target. But that heavy reliance on physical shape creates a severe vulnerability. Enzymes are incredibly sensitive to their surrounding environment. When the environment gets too cold, the surrounding molecules move sluggishly. The substrates simply don't bump into the active sites often enough to keep reactions running efficiently. High heat is even more dangerous. If temperatures rise too much, the intense energy causes the enzyme protein to unravel and melt out of shape. It loses that precise active site structure, permanently ruining the lock and key fit in a process called denaturation. Acidity levels represent a second critical environmental factor. Enzymes require a specific pH, a Goldilocks zone, to maintain their structural integrity. Take pepsin, an enzyme whose job is breaking down proteins in your stomach. This chart illustrates pepsin's activity levels. It operates perfectly here at the highly acidic pH of 2, the exact environment of gastric juices. But as the scale moves toward neutral pH 7, the activity level drops to 0. It becomes inactive. Finally, the speed of any reaction is limited by concentration. The ratio of available enzymes to substrates sets a hard biological speed limit on how much work can be done at any given moment. Your body constantly expends massive amounts of energy regulating its core temperature and balancing internal pH. It does this precisely to protect these delicate microscopic machines from breaking down. Zooming back out from those microscopic pockets to the scale of the whole human body, the scope of what enzymes accomplish becomes clear. Millions of these precise lock and key reactions are executing inside you right now. It's a high-speed, incredibly complex biological network operating silently in the background of everything you do. Yet this entire system balances on something as fragile as proteins, maintaining a perfect geometric shape. Without these delicate catalysts working to bypass the slow pace of natural chemistry, human biology would simply grind to a halt.